Good afternoon, everyone. Welcome to our Muscle Health Research Center seminar today. My name is David Hood. It's a pleasure to introduce our speaker today, Dr. Douglas Millay from the University of Cincinnati, Cincinnati Children's Medical Center, of course, in Cincinnati, Ohio. Doug is originally from Kentucky and went to Northern Kentucky University, and then worked at the University of Cincinnati, where he did his PhD with Jeff Malkenton on calcium regulation and muscle contraction. Then spent 5 years in a laboratory with Eric Olson at UT Southwestern in Dallas. Did some fascinating work on myoblast fusion, discovered some very, very interesting proteins, myomaker and myomixer, and really set that field on fire, I think, understanding myoblast fusion and muscle formation. Today, he is going to talk about multinucleation and myonuclear accretion during muscle adaptations. Really looking forward to this talk. And so, Doug, if you don't mind, share your screen and unmute yourself, and let's take it away.
Sure. Thanks, David. Let's see. Everything look okay?
Yep, it does. Very good.
Thanks, David. I really appreciate the invitation. Nice introduction, the opportunity to present our work. So obviously it's unfortunate we can't meet in person, which would also allow me to learn more about the work at your center, but I'm looking forward to any feedback on our work. And so what I thought I'd do today is to tell you about our work on the role of multinucleation and myonuclear accretion in the control of skeletal muscle development and adaptations in the adult. And so what I'll tell you about is, many of you know sort of the need for satellite cells and myonuclear accretion it's been a little controversy in the field. And so I'll give some history on the topic and explain that I think actually both sides can be right. And also talk more than just about the need for myonuclear accretion and tell you about a surprising discovery that, myonuclear numbers themselves may regulate myonuclear domain flexibility, which are determinants of myofiber size and growth. And so many of you know that muscle comprises approximately 40% of our mass and is necessary for breathing, and locomotion is a major site of metabolism. And it's also an incredibly important tissue to maintain our quality of life during aging, and also in response to sort of chronic diseases. And the cells that make up skeletal muscle are skeletal myofibers, and they're one of the largest cells in our body. So here's an EDL from the mouse, which is about 5,000 microns in length, whereas a cardiomyocyte is about 100 to 200 microns in length. And so, in fact, in humans, muscle can be up to 42 centimeters. So it's one of the largest cell types, even in humans. And the way that muscle deals with its large size is through multinucleation. So it's uniquely multinucleated. And so in this EDL myofiber, there's about 220 nuclei in one cell. And so we got interested in multinucleation through our discoveries associated with the molecular control of myoblast fusion. And so during muscle development, myoblasts, or stem cells, are specified to the myoblast lineage through MyoD and myogenin. And these factors are also important for differentiation of these cells to a multinucleated myofiber. And what was sort of unknown prior to our work was how myoblasts govern the fusion process in a muscle-specific manner. So what we did is we discovered two factors that are only expressed in muscle that really drive the fusion process. So the first one is myomaker, which I discovered during my postdoc with Eric Olson. It's a 7-pass membrane protein that's necessary for fusion. And then the other protein that's required for fusion is myomerger, which is an 84 amino acid protein that I discovered in my independent lab, co-discovered by also Eric Olson's group and a group at Novartis, and they named it myomixer and minion respectively. But sort of the the key piece of data that just shows how central these factors are for the fusion process is that when we genetically delete either myomaker or myomerger, there's a complete lack of muscle formation. So here are embryos of myomaker. You can see bones. There's absolutely no muscle around them, and the same is true for myomerger. And so we were interested to study these factors and study how they might regulate fusion. And so what we were really interested in is that sort of robust myonuclear accretion occurs during postnatal development. And so here is a postnatal timeline in the mouse where you can, and it's plotted with myonuclear number. So you can see the myonuclear number increases in the mouse sort of linearly from P0 to P21. And then it's sort of, there's a steady state that's happened. There's some debate on whether myonuclear accretion here, it likely does. There's likely myonuclear accretion that happens, but it's certainly not as robust as what occurs during development. And so we were fascinated by the idea that muscle accumulates all of these nuclei during development. And not only does muscle develop with hundreds of nuclei, but it can also acquire nuclei from the stem cell pool throughout life. So muscle has the presence of these muscle stem cells or satellite cells. And when you exercise, these cells get activated to fuse and increase myonuclear numbers in a muscle fiber. And so, so while there are other cell types in a body that also require multinucleation, such as osteoclasts or placental trophoblasts, muscle is the only one of these cell types that sort of lasts throughout the lifespan of the organism. And so we think there are important regulatory mechanisms that control these old and these new myonuclei function to maintain muscle. And so my lab began trying to elucidate the mechanisms of myoblast fusion through studying myomaker and myomerger. And then we started to think about, what are the consequences of fusion? And obviously the consequence of fusion is that the ultimate goal is to provide new nuclei for myofibers. So it got us thinking about the purpose of multinucleation in muscle and how this regulates muscle size and adaptations. And so we started to think about what are the potential advantages of multinucleation in muscle. And so I think we came up with a couple ideas, and there's potentially more, but two I think are the most profound ideas are, first is that nuclei within this large syncytial cell can only control a finite volume of cytoplasm. So it's sort of the myonuclear domain theory. And then the other idea is that there are specialized regions that require transcriptional diversity. And so we know that's true in at least two areas of muscle. So one being underneath where the nerve innervates muscle, the nuclei there are transcriptionally unique, referred to as the neuromuscular junction nuclei. And then at the ends of myofibers where tendons attach, the nuclei there are also transcriptionally unique. And so what we wanted to do was we wanted to test this idea of transcriptional diversity. And so we needed some new tools to be able to do that. And so what we did is we developed a single nucleus RNA-seq strategy. And so what this is an ability to detect transcripts from individual nuclei within all of muscle. And so what we did is we took a wild type mouse, we dissected its tibialis anterior, we isolated nuclei, and we fax purified these nuclei. And then we used 10x chromium to barcode these individual nuclei. And we performed this single nuclear RNA sequencing at multiple time points throughout the lifespan of the mouse. And we did find transcriptional diversity during development and aging. And so here's a UMAP projection of postnatal day 21 and 30 months of age. And so if you're not familiar with single nuclear sequencing, each one of these dots represent a nucleus. And so what we can do is we can categorize these nuclei based on expression. And we observe all the cell types that we would expect to see in muscle. So we can find satellite cells, fibroadipocyte progenitors, endothelial cells, immune cells. And we can also detect the myonuclear population, so type 2X and type 2B. We can detect the rare myonuclear populations that exhibit transcriptional diversity, such as neuromuscular junction and myotendinous junction. And so you can tell at 21 and 30 months of age that there is some transcriptional diversity that we see in these sort of ENA, NOS1 populations and ENA populations here that don't sort of just categorize based on fiber type. And we think those populations might be important for sarcomere assembly. But when we look at sort of homeostatic muscle at 5 months of age, there's more of a continuum of transcription and not so much diversity. And so we think that the transcriptional diversity that we fail to detect at baseline sort of suggests that it doesn't fully explain the level of multinucleation that we observe in skeletal muscle. And so incidentally, we've shared all of this data with a website where users can go and sort of input their favorite gene, and you can see the expression of that gene in the various populations of wild type skeletal muscle at post time points from P10 to adult time points to P30, or so 30 months of age to the lifespan of the mouse. And so this sort of takes us back to the conventional explanation for multinucleation, which is the correlation between DNA content and size. And so this correlation is conserved in almost all cell types, even in humans where the cells, so this is a plot of DNA content per volume, and the cells with the larger volumes are associated with larger amounts of DNA content. And in muscle, shown over here, this relationship is referred to as the myonuclear domain theory, where each nucleus is predicted to control a certain volume of cytoplasm within the myofiber. And so the idea of a finite sort of MND volume has commonly been interpreted to mean that the sort of intrinsic biosynthetic capacity of a myonucleus in healthy adult myofibers is fully utilized and limiting. But we're going to explore this assumption in the talk today, as it directly, I think, relates to the regulation of myofiber size and adaptations. And so this myonuclear domain theory has been led to questions in the field that have sparked, a lively debate for, I would say, the better part of 20 years. And some of these questions that have been discussed are, is there a myonuclear domain ceiling, or do myonuclei exhibit a transcriptional reserve? Can the existing myonuclei do more? Does adult muscle require new nuclei for growth? And does myonuclear accretion occur for reasons other than growth? And so what I'll tell you today is the existence of sort of myonuclear reserve capacity, transcriptional reserve. It doesn't necessarily negate the requirement for new nuclei. And so I think the presence of both myonuclear domain flexibility and the need for myonuclear accretion can both be true. And so that's sort of going to be the theme of the talk today. And so in terms of myonuclear domain flexibility, we already know that this is possible in certain contexts. And so one context is postnatal development, where I told you that during development that there's a lot of myonuclear in the mouse. There's a lot of myonuclear accretion that occurs between P0 and P21. But if you consider the amount of growth that in cross-sectional area that occurs from P21 to the adult, shown here in the cross-section of a TA, you can see that there's a large amount of increase in size independent of increases in myonuclear number during the same time frame. So during this time, the myonuclear domain grows in a large amount. And we also know, in response to pharmacological activation through the beta-adrenergic agonists or myostatin inhibition, that you can drive hypertrophy in adult myofibers independent of satellite cell activation. Satellite cells are activated, They don't fuse to fibers. And so that growth is just due to what's happening in the myofiber. But sort of, I think the real debate relates to physiological hypertrophy in the adult. And traditionally, this has been studied through the model of synergist ablation or tenotomy, where you would remove or cut the tendon of the gastroc, which is the lower leg muscle in animals. And so that would leave all of the load on the smaller muscles, such as the plantaris. And so you would get a large amount of hypertrophy 2 weeks after that stimulus. And sort of the one thing that's agreed upon in the field, I think, is that satellite cells are indeed activated in these models, and they fuse to fibers in giving those fibers more myonuclei. And so what the field has done is we've sort of started to manipulate satellite cells in the context of this synergist ablation. I just listed sort of the studies that have done this. And so some studies have looked at short-term time points after synergist ablation and assayed whether activation of satellite cells and myonuclear accretion was needed for hypertrophy. And so the Peterson group at the University of Kentucky showed that there was normal myofiber hypertrophy when they deplete satellite cells after 2 to 3 weeks. But then when they look longer, shown down here, that they also showed in a FASA paper that you sort of need these for more long-term hypertrophy. And then other studies from the Gunderson lab, my lab, and then the Soteropoulos lab also showed that at short-term time points, you do need satellite cell fusion for hypertrophy. Fucata lab has had some really interesting papers recently showing that you do need satellite cells for long-term hypertrophy as well. And so recently, there's been development of more physiological models of hypertrophy. So the Peterson lab developed Power, where they add weights to a running wheel to model resistance exercise, and ablation of satellite cells there also results in blunted growth. And then my lab developed a high-intensity interval training protocol that we call HIIT. I'll tell you about some of that data today, where we delete myomaker, so we block fusion in stem cells, blocks myonuclear accretion, and we don't get proper muscle adaptations. And then Troy Hornberger's lab at the University of Wisconsin recently developed a really cool weight pulling exercise regimen for mice. And so I think that's going to be an important model to study in the future as well. So based on the data that I presented in these last 3 slides, where we know myonuclear domains can be flexible, and we know that the majority of data suggests that, myonuclei need to be added to muscle for growth, I think that we can come up with a unifying statement, that myonuclear domains are indeed flexible, but there is an obligatory requirement for myonuclear accretion for sustained growth. So I think this is important because it's been argued that the existence of one sort of negates the other. And I think that doesn't have to be true. I think they both can and do exist. And so I think there's more critical questions that we're interested in investigating, regarding these questions. And one of those is that, when is the regulation of existing nuclei not sufficient to adapt, and myofibers then require new nuclei from the satellite cell pool? What are the mechanisms that control myonuclear domain flexibility versus accrual? And then what are the new myonuclei contributing to this incision? And so what I'm going to tell you about today is sort of the beginning of our efforts to answer these questions. And most of the advances I'll tell you about today related to this question of a mechanism that impacts myonuclear domain flexibility. And so what we wanted to do is we wanted to be able to manipulate myonuclear number, because many studies, especially during developmental growth, many studies have been correlational. So they just look at the number of nuclei and how that correlates to size. But we really wanted to manipulate myonuclear number. And the way that we manipulated myonuclear number is we developed this fusion incompetent mouse model. And so I told you this gene myomaker is absolutely essential for the fusion of muscle stem cells to the myofiber. And so what we did is we crossed a myomaker allele that where the gene can be deleted in a temporal manner, specifically in satellite cells. And we do that with a satellite cell specific Cree PAC7R. So we give the mice tamoxifen, it renders all of the muscle stem cells fusion incompetent. And so the ability for this mouse to ablate fusion is shown in this experiment on the right, where we injure the mouse after tamoxifen treatment. You can see in a control that there's robust regeneration. You get centrally nucleated myofibers, but when we ablate myomaker and stem cells, there's no fusion and no regeneration that happens. And so we began by asking if myonuclear number is the primary determinant of muscle size. And so what we did is we used these fusion incompetent mice and we induced deletion of myomaker at postnatal day 7 in the mouse, because this is at a time point when there's robust myonuclear accretion. And we refer to these mice as delta 2-week mice because they become fusion incompetent at some point in the 2nd week of life. And so this method works extremely well. So we're able to precisely titrate the number of myonuclei. And so when we treat the mice with tamoxifen at P6 and we ablate fusion, and we analyze 4 weeks later at P35, what we see is about a 50% reduction in the number of myonuclei. So here's a control EDL myofiber, has about 220 myonuclei, and then a delta 2-week myofiber, you can see has about 100 myonuclei in those myofibers. So there's about a 50% reduction of myonuclei. And so the first question that we wanted to ask is what happens to myofiber size when you reduce myonuclear number by 50%? And so here is a cross-section of a control TA as well as a delta 2-week TA. And you can see that there is a reduction in myofiber cross-sectional area. And this is quantified here. It's about a 17% reduction. And when we look at the volume, not just the cross-sectional, but the entire volume of an EDL in these two groups of mice, you can see a 34% reduction in the delta 2-week mice. And so this sort of reveals, I think, an important concept is that there's not a one-to-one relationship in terms of the number of nuclei and size, that sublinear relationship. And this was also sort of shown in a companion paper in Nature Communications with Christian Gunderson. He looked at scaling relationships in human myofibers. But it's important to note it's not a one-to-one relationship. And so we next wanted to know whether this increased muscle size was associated with larger myonuclear domains. And so this was done by Christian Gunderson's lab in the University of Oslo. You can see that this is a segment of a control myofiber and a delta 2-week myofiber, 3D rendering. So you're looking down on the myofiber, and myonuclear domains were quantified from these areas at multiple postnatal ages from P14 all the way to P150. So you can see increases in EDL myonuclear domains beginning at P28 and lasting until the adult. And so there are increased myonuclear domains in this delta 2-week muscle. And so one thing that's important to note about expanded myonuclear domains is that larger myonuclear domains aren't always functional. And so I think the best example of non-functional large myonuclear domains might be in organisms that have mutations in myostatin. So here's a Belgian blue bull. You can see how jacked it is. It has huge muscle. But if you actually look at its specific force, there can be reductions in specific force because not the entire amount of cytoplasm within a myofiber is not necessarily made up of functional sarcomeres. And so we wanted to test whether there were larger myonuclear domains in these delta 2-week muscle. And so what we did is we collaborated with the Sadi Yapan lab at the University of Cincinnati to do muscle force. And so we measured the force, peak force for both control and delta 2-week mice, and this is in the adult at 5 months of age. And so what we can see is a reduction in peak force in these delta 2-week mice, which makes sense because the overall muscle is smaller. We normalize this, this force to muscle size, and we obtain a specific force measurement. Now you can see that the delta 2-week mice is a sort of equal to the control. And this is at a per given area that the delta 2-week mice is generating the same amount of force as control. And so then we started to think about what could be controlling these larger myonuclear domains. And since the major manipulation in these delta 2-week mice is just that they have fewer myonuclei, we wondered if the existing myonuclei might be functioning at an increased capacity. And so what we found is that there is increased mRNA content per nucleus in delta 2-week muscle. And so what we did is we did a microarray comparing wild type, and which is shown down here on the x-axis, and delta 2-week mRNA shown here on the y-axis. And what we did is we normalized the signal for each gene that we obtained from the microarray. We normalized that to myonuclear number from that particular sample. And what we see is we see a slope that is a 3.5, suggesting that for every given mRNA, there's a higher content per nucleus in the delta 2-week compared to control. If it was 1-to-1, we would expect there to be a flat slope. And so, so I think what this suggests in our first study here is that a transcriptional reserve capacity does exist to support expanded myonuclear domains in functional growth. And that's observed in these delta 2-week mice that have increased myonuclear domain size and functional myonuclear domains. And so this sort of leads us to the question of whether adult myonuclei possess the same transcriptional reserve capacity that would be sufficient for exercise-induced adaptations. And so, I think before that, during exercise, that muscle stem cells can get activated and fuse to the myofiber and supply those nuclei. And so what we wanted to first do is we wanted to develop a more physiologically relevant model of hypertrophy. And so, versus synergist ablation. And so what we did is we developed a high intensity treadmill protocol where we put mice on a treadmill, and then we would adjust the speed or the incline over 8 weeks. So they were progressively doing more work. And at the end of this 8-week protocol, you can see, there's an increase in muscle size as well as increase in muscle force, suggesting that it is increasing hypertrophy of muscle, or driving hypertrophy of muscle. And so what we found when we looked at myonuclear accretion was that when we looked at multiple time points after this HIIT protocol, that we can see a progressive increase in myonuclear number. So that myonuclear accretion does not occur at just one time point. It occurs at multiple time points, multiple stimuli after or during HIIT. In terms of muscle size, what we see is that muscle size occurs sort of late in the protocol. So here's a cross section of muscle at 2 weeks, 4 weeks, and 8 weeks. And when we evaluate myofiber size, we only see significant increases at 8 weeks of the protocol, but not at preceding time points, even though myonuclear accretion is occurring throughout. And so then we wanted to test whether you need muscle stem cell and myonuclear accretion to adapt to this HIIT protocol. And so what we did is we took these adult mice, 6 to 8 months of age, and we hit them with tamoxifen to make them fusion incompetent just at that time point. We subjected them to HIIT, and then we had sedentary mice as a control, and then we analyzed these mice during the protocol. And what we noticed in these myomaker satellite cell knockout mice is that they become progressively exercise intolerant. So this is the number of times that the mice will visit the shock grid that's below the treadmill. In the control and the myomaker satellite cell knockout mice, you can see that the fusion incompetent mice visit this shock grid at a greater frequency as they go throughout the protocol. And so we refer to them as exercise intolerant. And so this inability to deal with the exercise, we think the reason for that is the fact that there might be some damage early parts in the exercise protocol that aren't repaired in these fusion incompetent mice. And the data for that is shown here, where we can detect increased levels of serum of creatine kinase in the serum in the fusion incompetent mice at 2 weeks and 4 weeks after HIIT. Creatine kinase is a muscle factor that can be released into the serum due to membrane damage. And so you can see increased membrane damage in the fusion incompetent mice, and this leads to fibrosis. So when we look at fusion incompetent mice after HIIT, we can see an increased amount of trichrome staining, indicating increased fibrosis. So the fact that when we ablate muscle stem cells at the beginning of this HIIT protocol, that the mice don't exercise as well and they sort of have a maladaptive response, it sort of precludes the ability to test whether myonuclear accretion is needed for growth. And so what we really wanted to test is if there's a function for these nuclei that are added during the late stages of HIIT. So here what we do is we take these mice, both control and myomaker Flox Pac7 Creon mice, and we don't treat them with tamoxifen. We don't make them fusion incompetent. We either have sedentary mice or we expose them to HIIT. But 4 weeks into the protocol, then we treat them with tamoxifen, and we're inducing fusion incompetence in a group of these mice in the middle of the protocol. And so basically we're blocking fusion that would happen at the last 4 weeks. And so, so what we see here first is that these mice are able to exercise normally, so there's no exercise intolerance. But then when we look at muscle size, what we see is that these fusion incompetent mice that were induced at 4 weeks of HIIT, now they're no longer able to grow compared to the control. And so I think what these, I think an important insight from these HIIT studies is that myonuclear accrual is not utilized exclusively for growth. So what we observe is that during the early stages of HIIT, that there's myofiber damage, that addition of myonuclei occur throughout the entire protocol, but size is only increased at later stages of HIIT. So if we block fusion during the final 4 weeks of HIIT, there's not growth, even though myofibers have accumulated the nuclei during the initial weeks of HIIT. And so we think that you really need this myonuclear accretion for growth, but that it's not only myonuclear accretion happening for growth. So we think that maybe that myonuclear accretion happening here is maybe more dedicated to repair, whereas myonuclear accretion happening at later stages might be dedicated to growth, suggesting there might be specialized roles for newly fused myonuclei. And so to summarize this part of the talk, myonuclear accretion is required for exercise-induced adaptations, so response to damage and growth. And that adult resident myonuclei exhibit sort of minimal flexibility to deal with the required adaptations to HIIT. And so you may be thinking, well, we showed you before with the delta 2-week mice that myonuclei are indeed flexible, but that doesn't seem to be the situation in the adult. And so we were wondering what could the differences be between the adult and development. And so one of those differences, there can be many, of course, one of those differences is that in the adult, there's a lot more nuclei present in the syncytium compared to during development. So we wondered if myonuclei numbers themselves regulate this transcriptional reserve capacity and an ability to do more. And so what we did is we wanted to test this idea as we developed an additional model with fewer myonuclei than wild type, but more myonuclei than delta 2-week muscle. And so we went back to these fusion incompetent mice. And so instead of treating them with tamoxifen at P7, like we did with the delta 2-week mice, we treat them at P14. And we refer to them as delta 3-week mice because they become fusion incompetent in the 3rd week of life. And so what we've done with this model is we've generated mice that now has 180 nuclei per myofiber. So we treat postnatal day 13, and then we harvest 4 weeks later. And so here's an EDL of a control. It has normal 220, 250 myonuclei. And then these delta 3-week mice have about 180 myonuclei per myofiber. So we have wild type mice that have 220. We have delta 3-week mice that have about 180 nuclei per fiber. And then we have these delta 2-week mice that I told you about previously that have about 100 nuclei per fiber. And so what we did is we compared the size of the delta 3-week muscle to control and delta 2-week muscle. And so here's a cross section. If you compare the control TA to the delta 3-week TA, you can see in the delta 3-week, we have a 22% reduction in cross-sectional area. And in the delta 2-week, we have a 31% reduction. So somewhat sort of comparable levels. And what we really wanted to know is what is the transcriptional reserve capacity of these delta 3-week mice. And so now we turn to a qRT-PCR where we look at the levels of sarcomeric genes, so myosins, troponins, in both the delta 2-week shown in orange and the 3-week shown in purple. And these mRNA levels are normalized to the average number of myonuclei. And so what we can see is we can see a robust ability or robust levels of these genes in the delta 2-week muscle, suggesting increased mRNA levels on a per nuclear basis. And we see some increase in the delta 3-week, but much less robust compared to what we see in the delta 2-week, suggesting that these delta 3-week muscle or myofibers are not able to mount the same transcriptional reserve capacity compared to delta 2-week. And so I think this leads to sort of a couple summary slides where our data really say that myonuclear numbers and DNA content are the ultimate determinants of size, that myonuclei exhibit a transcriptional reserve capacity during development, and that transcriptional reserve capacity is regulated by the number of myonuclei. So I just want to show you this model here where we think there's an inverse relationship between myonuclear number and actual activity of the myonuclei. So as myonuclear numbers increase, the transcriptional reserve capacity actually decreases. And so we speculate that there might be a cost paid at the level of individual nuclei, where as their numbers increase, there's a progressive reduction in their ability to utilize this reserve capacity. And so I think it's very interesting to think about mechanisms by which this might be achieved in terms of as more nuclei are added to the syncytium, each one does progressively less. And also, I think, the data also show in the adult that there's an obligatory requirement for myonuclear accretion in the adult for sustained functional growth. And I think that is consistent throughout the majority of studies that have been published. And you may be thinking here that the data I showed you suggests that myonuclear domain flexibility in the adult doesn't exist. However, I just want to point out that human studies have shown that after resistance training, that smaller myofibers were able to support hypertrophic growth independent of an increase in nuclear content, whereas growth in larger myofibers were associated with an increase in nuclear. And so I think an explanation for this might be the idea of a myonuclear domain ceiling. And I've tried to schematize this for you here. So we know that myofibers in muscle, they're heterogeneous. Sometimes we have larger ones, sometimes we have smaller ones. And so if we think of there are myofibers in muscle with large myonuclear domains. So this red line is the myonuclear domain ceiling. So the myonuclear domains are sort of already at that ceiling. If you increase workload in these myofibers, the way that these myofibers need to adapt to that and be able to deal with the increased workload is that they have to add a nucleus because they're sort of at that myonuclear domain ceiling. But other myofibers within the muscle that have smaller myonuclear domains, they have some room to expand that myonuclear domain ceiling. And when they are subjected to increased workload, that they are able to increase their flexibility and their growth potential is reached through reserve capacity. And so, so we think that the myonuclear domain ceiling is consistent with the paradigm that myonuclear numbers are the ultimate determinants of size and the reliance of myonuclear accretion for growth. And so with that, I'd like to acknowledge the lab and the people that did all this work. So it's a super hardworking group. I'd like to acknowledge so multiple people here. So Chingo was a postdoc in the lab who did all of the HIIT work. Alyssa Wilson was a master's student in the lab that did a great job with neonatal development work, the Delta 2-week, Delta 3-week mice. Michael Petrani and Casey Swoboda here did the single nuclear sequencing work. And then a bunch of collaborators that I need to thank. Vikram Prasad, who is in the Multinucleation lab at Cincinnati Children's, Christian Gunderson, University of Oslo, Saktivil Sadyapan did some functional stuff for us. And then of course, all our funding sources. So with that, I'd be happy to take any questions.
Doug, thank you very much. That's outstanding work, really fascinating tools and techniques that you've got going there. And it's such a hot topic and so controversial in the field. And you really showing some nice data. So
I think I was trying to say it's maybe not as controversial as everyone may think. I think both things can be true. Myonuclear domains can be flexible, and you need myonuclear accretion for sustained functional growth.
Right. So folks, we'll take questions in the Q&A, and I'll get to those.
Should I stop sharing my screen or?
Yeah, you can. You can stop sharing unless somebody wants to see a slide or two, but perhaps not. Let's stop sharing, and we'll just take some questions. I'll start before I go to the Q&A. Folks, if you want to ask some through the Q&A, please do so. It seems like, when you talked about a ceiling for myonuclear domains, you talked about large myonuclear domains not actually being functional. And you said something about the Belgian blue cow as an example of that. And to me, that points to a ceiling. Doesn't it point to a ceiling? It says that the large, as you expand the myonuclear domain, you get to a certain point where the amount of mRNA, but the amount of gene expression per nucleus is no longer sufficient to make a good quality muscle fiber.
Yeah, I think, yeah. So let me clarify a couple things there. So first, the point of mentioning the Belgian blue cow and myostatin mutations is that the point was that not all larger myonuclear domains are functional. So it's really important to not just look at the size of myonuclear domains, but try to evaluate the function of them. But yeah, I agree. I think that that's sort of the paradigm, is that the fibers that are already sort of maxed out in terms of their myonuclear domain, those are the ones that probably need to add a new nucleus through myonuclear accretion to be able to grow and adapt. While there might be other myofibers that have some, haven't reached their myonuclear domain ceiling, and they have some transcriptional reserve capacity. So those could grow, through activation of the existing myonuclei. And that's why I think, the questions of when and how myonuclei activate that transcriptional reserve capacity versus requiring a new nucleus, I think are the most pertinent, now in the field.
Yeah. And the other thing related to that is you talk about a reserve capacity, but I'm wondering about the signaling. Now you say, as you increase the number of myonuclei, the reserve capacity goes down. But I'm wondering if the signaling is to that, because you're measuring that as mRNA, are you not? And I'm wondering if the signaling is reduced if you have more myonuclei. And so it's not really reserve capacity, but it's actually what the nucleus is putting out in response to the signals. If you have more myonuclei, perhaps the signaling toward transcription is reduced.
Yeah, I think what you're getting at is that we really have no idea what signals or what's controlling this reserve capacity, or or the activity of myonuclei within a syncytium. And I think that it's really hard for me to answer your question without sort of knowing. And I think there's a couple things I could speculate on in terms of those mechanisms. One might be that maybe that there are some limiting transcription factors in muscle. And so the more nuclei and the more templates that you add to that syncytium, it's more difficult for those factors to activate transcription in each of those nuclei. The fewer nuclei you have, those transcription factors are more, let's say, more efficient. But it's pure speculation. But I think it's a really cool question, not only in terms of muscle biology, but any sort of cell type that is multinucleated.
Right. Okay, so some questions from the Q&A. Ola Adagoki asks, says, thank you for a great talk. In the myomaker deficient animals, there was presumably satellite cell activation in response to exercise. What happens to these cells that did not fuse?
Yeah, that's a good question. So we've done a little bit of stuff on this. So they certainly activate. I think some of them may die. We have some data, in our muscle overload study that they're still maintained, but we also have some data that I think maybe is unpublished during development, where in the myomaker knockout animals, you can see more tunnel positive cells and things like that. So some of them may die. The short answer is that we don't know exactly what happens to them. We've not sort of taken them out or lineage trace them and see anything like that. So we're not 100% sure, but we do know that they're activated and that some of them stay around.
Okay. Stephen Alway asks, very nice talk, Doug. Your work has been largely in type 2 fibers. Do you know if the ceilings for type 1 fibers would be similar or different from type 2 fibers?
Yeah, so it's a great question, and it's something that we've done a little bit of. So in like the Delta 2-week mice, we looked at the uh, soleus, and we saw sort of similar effects in terms of larger myonuclear domains and increased transcriptional reserve capacity. But we haven't done a proper comparison and compare the magnitude of that or the limits of that between type 1 and type 2 fibers.
Okay. John McDermott says, thanks for the talk, Doug. The TA muscle is a mixed muscle. Can you comment on whether myonuclear accretion or reserve capacity vary depending on the fiber type? I guess it's a similar question to what Dr. Alway asked. Yep. Okay.
Yes, we should do those experiments, huh?
Yeah. Yeah. There's a lot of interest in fiber types, and fiber type responses, of course.
Sure.
Yeah. Anthony Shime asks, have the opposite type of experiments with a mouse model for increased availability of myoblasts or myoblasts with greater fusion capacity been performed by you or others?
It's a great question. The answer to that right now is no, but it would be super interesting to learn about or figure out what the results of those sort of mouse models would be.
Yeah. Can I ask, do you believe that a nuclei turnover, like suppose you have a disuse model, or do you believe that in the myonuclear apoptosis and loss of nuclei with that kind of stimulus? Not everybody does, so
You're just hitting all the controversies, aren't you? Just hitting all of them.
I'm trying my best to hit all the controversial things.
So the first thing I should say is that we haven't really done any work on that at all. We don't have any primary data, so my comments would just be based on the literature that I've seen published. I think that there's some reports where they don't see nuclei getting lost, I think, and then there are some that they do. And I'm not really sure. Do I think it's possible? Sure, it's possible. I think that I think hopefully what the field does is they go beyond just saying whether it exists or not. And I think the key would be to show molecularly how, if those nuclei are lost, how they're lost, right? Is it like a reverse fusion event that the nuclei are spit out? Or they or is it nucleophagy or something like that? I think that is really, I think, the key to really knowing whether they're lost or not. And I think, there can be times when maybe you can see changes in nuclei number or reduction or maybe, some nuclei are lost, but it's a more rare event. It doesn't happen at physiologically relevant levels. So I guess to sum up my question is I'm on the fence. I'm not really sure. I think it would be cool if we could figure out, mechanisms by which they could lose those nuclei.
Agreed. Good answer.
And not just whether they are there or not.
Yeah. Good answer. So just to, get Charlotte Peterson involved here perhaps and say, she would probably say, and her group would say, myonuclear accretion is not required for adaptations to exercise. And you've concluded that myonuclear accretion is required for growth and adaptations to exercise. So where do you, and I understood your point about myonuclear accretion is required for long-term growth and stability, I suppose. Is that where your opinions might differ?
I actually don't think so. I think they have data too. I mentioned it in their, I think, 2014 FACET paper where they looked at longer times after hypertrophy and showed that when they ablated satellite cells that the muscle doesn't grow as well. And then they also see in their power model where you need myonuclear accretion for adaptations associated with that. So I think that there's more alike than there is different if you sort of take a step back and look at all of the data overall. And, they have really interesting work about sort of non-fusion roles of muscle stem cells, which is certainly possible. I don't think those non-fusion roles negate the importance of myonuclear accretion at all. I think both of those can be true. I think a lot of biology that like it's almost never black and white, right? There are these nuances, and I think there's for long-term sustained hypertrophy, I think that their data, our data, the field's data is in agreement. And maybe what the difference is in those short-term time points might just be the magnitude of hypertrophy that was induced. And so that, there's differences between those short-term experiments between labs.
Yeah. They also use the ablation model, and you've got an exercise model going on there, so different models as well. But yeah, I can understand that both could exist. Okay, ladies and gentlemen, any more questions for Dr. Millay? Seeing none popping up, Doug, stay with me here. Thank you very much for your talk. It was great. Sure. And thanks everyone for coming and attending today's colloquium session with Dr. Millay. Seminar is over, and we'll see you next time. Stay with me, Doug.
Sure.
Oh, good. Okay, we're done. Yeah, sorry about that. I hope I didn't, I hope I didn't, let me just turn off this recording here.